Thank you so much for the invitation. It's such a delight I think this is my first conference in 2 years. And after so much isolation, it's a delight to hear people talk about their work. So today I wanted to share some initial insights into an overarching problem and project in my group. So I started my group at Carnegie Mellon a few months before the pandemic started. And overall, we studied the spatial architecture of cellular and molecular and organismal biological systems and ask how the spatial architecture affects the system's rate of evolution. And I want to start on this kind of more general slide, especially for the students in the audience, to restate the fact that even though we know the basic rules of evolution for a long time, since the time of Darwin, we still lack so much mathematical formalism, and there's still so many unknowns in evolutionary theory. And I think it's still such an exciting time to study evolution because we still have to figure out so many things, from something as fundamental as the meaning of fitness to a more general question like how many routes to higher fitness, what determines rates of evolution. And in particular, I've worked on a bunch of different questions that span some of the ideas on this slide. But for the past couple of years, I focused particularly on understanding how spatial structure and evolutionary barriers shape rates of evolution. And the reason for that is because through recent innovations in imaging techniques and the ability to process these images at scale, we have absolutely unprecedented access to public data sets of spatial organization of molecular and cellular systems. And in fact, modern molecular research increasingly relies on high-resolution image data as fodder for innovation. However, I believe these data sets are underutilized in evolutionary biology. And in order to make full use of them, in order to understand how the evolutionary processes have shaped these patterns of organization, how these static patterns, how the snapshot in time will shape future evolutionary dynamics, we need to go beyond what is currently done, which is simple description of pattern, towards understanding something about function. And that means building models to understand the role of space in evolution. And we're by no means the first ones to ask about the role of space in evolution. There's so much work already on it. And that's why, when I was thinking how many citations should I put on the slide, I realized, there's so much work already. I would miss someone's work. And there's a ton of previous modeling approaches. However, most previous modeling approaches represent spatial structure as a small number of demes or patches, right? So the first easiest thing to do is to take a well-mixed population and divide it into two, and you have your two demes or two patches. And these small number of demes or patches are then connected by migration corridors. And these simple topologies are great for analytical tractability, for us understanding mathematically and intuitively what's going on in the system. However, these more tractable models are hard to use and to link to these data sets, to these novel data sets that are messy with large amounts of spatial heterogeneity, complex patterns of localization and co-localizations. How do you link something like, a very simple, 5D model to the mess, to the spatial mess that we're seeing in real data sets? And I'm going to give an actual application example a little bit later. The problem, though, is that studying more complex topologies, going beyond these simple descriptions, is a lot harder, right? And so the challenge for this problem, if we want to link mathematical models to novel imaging data sets, the challenge lies in finding just the right spatial geometric representations that can capture the complexity, but that also are analytically tractable and intuitive. And I don't have to sort of justify to this audience here why we would want mathematical modeling tractability. Now, one way to do this, and this is just an initial way, we are working on other approaches, but one way to do this, which is a very intuitive initial way, is to think of networks, to take these sort of homogeneous, deemed connected by migration corridors, and instead don't connect everything. Think of the structure, use the structure of a network where the nodes could be individuals, or they could be homogeneous, some populations of some sort, and the links represent the local patterns of interaction and replacement. And the links connect the nodes and allow the mutation to spread through the population, right? And we can use the evolutionary formalism of the Moran process on these graphs and then ask, how does the structure of a network affect the spread of the new mutants? So in order to understand the role of complex spatial structure, we use this structure of a network. And just to sort of make sure that we're all on the same page when it comes to the rule of the stochastic process, so we have these individuals on a network, and then at every time step, if we do a birth-death dynamics, for example, we select a birth for death, select a node for birth proportional to fitness. Then we select a neighbor for death. And this is sort of the crux of how this local pattern of interaction contributes to the evolutionary dynamics. And this is where it would be different for a well-mixed population, because in a well-mixed population, you'd select a neighbor for death from the entire population. And then you replace the node selected for death with the offspring of the node selected for birth. So this is the stochastic process. And these types of network approaches have been used before in population genetic theory. Most of the work has been to study evolutionary game theory, where the nodes of the networks represent individuals playing games against each other. But initial work looking at how the spatial structure, as is modeled by the network structure, or I shouldn't even say the spatial structure, the structure of interaction and replacement, as it is modeled by the network structure, how does it affect proxies of evolutionary dynamics, such as probabilities and times to fixation? And here's, for example, one previous approach to this problem, where on the x-axis, we have mutant fitness. On the y-axis, I don't think my mouse works, but that's fine. On the y-axis, we have the fixation probability. And what this is from the Laura Henderson-Alney Trelson paper in PLOS Bio. And what the authors saw here, these initial preliminary studies show that for very small, notice the n equals 6 here, so 6 node structures, very small regular networks, we can compute these fixation probabilities. They don't change the fixation probability compared to well-mixed population that much. And in fact, one of the first general results here is the isothermal theorem that states that essentially graphs where nodes all have the same degree don't change fixation probabilities compared to well-mixed populations. So a bit of a letdown that we don't really see these large effects of population structure, especially given how hard it is to mathematically make progress when it comes to evolution on these networks. So for example, in Henderson and Trelson have this approach that is a based on the adjacency matrix to analytically compute these fixation probabilities. So the adjacency matrix tells us which nodes are connected to which, and it uniquely identifies the network. The problem with this approach is that you kind of have to keep track of a Moran process with 2 to the n number of states to figure out where the mutants are located on each of the nodes. And so even though the error rate for this approach is very, very small, the building time and solving time grow exponentially in population size. So it's really hard to get results for anything higher than, so here they say n equals 23 is kind of their limit. Another approach was this approach from Ben Allen together with Martin Novak that reduces the problem from exponential to polynomial complexity and population size by essentially figuring out sort of results in the limit of neutrality. So very, very small selection. And I think this is a great approach. The problem with this approach is it doesn't work for any significant amount of selective benefit of the new allele. So here, ignore kind of what's on the x-axis is the variance of the degree distribution. The y-axis is the probability of fixation. And we plotted sort of some of these approaches with lines, and then the dots are simulations. And it just doesn't quite work as you increase the power of selection. So we took a crack of this problem with some very promising results. And our approach, and the work was mostly done by my student YP, our approach uses the properties of nodes and edges of the network. And properties of the nodes are the degree distribution, which, as opposed to the adjacency matrix, doesn't uniquely identify the network. You can have the same degree distribution apply to very different types of networks. So it's not a one-to-one. However, by using the degree distribution, and I'm going to mention a little bit later also the mixing pattern, a property of the edges, we're able to make significant analytic progress into this problem. And this problem gets much, much simpler because instead of tracking the mutant at every node in the network, instead we group the nodes by their degree distribution and track frequencies of the mutant across these degree distributions. And then we essentially use a diffusion approximation. And this sort of proxy of the degree distribution for the network greatly simplifies our analytic approach, and it allows us to make progress for much larger and more systematic progress across any type of network from any network family. So just to convince you, before I go into sort of some of the intuition behind the results and some of the results that we're seeing, just to convince you that our analytic approach works, even though we're using a diffusion approximation, as we plot, let's say, the strength of selection on the x-axis, probability of fixation on the y-axis, our approximation matches the circles, the results of the simulations very well. So what do we see, right? This work is all pre-printed, it's all online, so I'm not going to go any further into the actual mathematical kinks because I really want to tell you about the results. So what do we see, right? We have this approximation, we can do simulations, and what we see is that, let's say we take an important network parameter like the mean of this degree distribution, right? This is one important sort of property of a network, and we plot it across from probability of fixation on the x-axis, and we see that as we increase the mean of the degree distribution, you approach a well-mixed population. So the more you connect things, the more you approach a well-mixed. That's all great and intuitive. And in fact, moreover, we see that, for example, in this case, the probabilities of fixation on average are on top of the well-mixed line. And researchers have actually defined these graphs that are above the well-mixed line for a beneficial mutation. And remember, we always use the well-mixed line as a null. The well-mixed line is a perfect null, and any departure from the well-mixed line gives us information into the role of spatial structure, because that's the only thing we're changing. So a graph is called an amplifier of selection if, compared to this well-mixed line, this well-mixed population model, advantageous mutants have higher fixation probability on this graph, and disadvantageous mutants have a lower fixation probability. So in a way, in these amplifiers of selection, the balance tilts towards selection and against drift. And it boosts, it's almost like beneficial mutations have an extra oomph, an extra boost in these amplifiers, and disadvantageous mutations are even more disadvantageous. Now, it turns out that it actually matters how you do the stochastic process on this graph. So let's say that instead of doing birth first and death second, let's say you do a death-birth dynamics, where first you select a node for death randomly from the population. Then you select a neighbor for birth, again, the locality, you select a neighbor for birth proportional to fitness. So now fitness, selection for birth proportional to fitness is local as opposed to the birth-death dynamics. And we replace the original node selected for death. You're already in the description of the birth-death process, you're already noticing this local versus global force of selection. When do we select proportional to fitness? And you can already intuit maybe this will lead to different dynamics on this network. And in fact, as we do the death-birth simulation, we see that for the exact same networks, right? For the exact same networks with the exact same properties, the birth-death versus the death-birth in green, so blue versus green on this plot, act very differently, right? And the green line is now underneath the well-mixed line, and a graph like this that for an advantageous mutant has a lower fixation probability than well-mixed is called a suppressor of selection. So here the balance of forces tilts against selection and towards drift. And in fact, there have been previous studies that say that most random graphs are amplifiers for birth-death, so the blue line here, but suppressors for death-birth. So we see some of the same results that Arnie Trelson and Laura Henderson saw before. Now, as we move away from these simple graphs, small n population sizes, and as we try to understand and to build a systematic theory across any possible graph type, what would be really useful would be to take different network properties to try to understand the evolutionary role of different network properties by systematically decoupling them and varying them one while keeping others constant. So for example, I want to vary the variance in degree, but keep the mean constant. I want to vary some kind of mixing pattern, but keep the variance and the mean constant. And this is actually not a trivial thing to do, but my student YP managed to build some of these algorithms using all kinds of edge swap algorithms to tune graph characteristics and almost allow us to traverse across network families while keeping graph characteristic constant in order for us to really understand the evolutionary role of each one and to really be able to build the systematic view into what properties exactly lead to amplification or selection. So using these algorithms as well as our analytic approximation. So whenever I have a line on a graph, it means analytic approximation. Dots are results of simulations. We're able now, for example, to keep the mean constant in this graph and vary the variance of the degree distribution and plot probabilities of fixation and try to understand how the variance of the degree distribution now affects these probabilities of fixation. So we see, for example, that amplifiers amplify even more with large variance in degree, and the reverse is true for suppressor. And in fact, the power of the analytic approach also lies in the fact that you are able to find sometimes, not always, but sometimes, these parameters that give you intuition into the dynamics. So, for example, for these death-birth, for the death-birth process on graphs, we're able to identify the selection amplification parameter, the parameter that essentially tells you the departure from well-mixed when it comes to probabilities of fixation. And this parameter for the death-birth process depends only on the mean and the variance of the degree distribution. And as we plot the selection amplification parameter on the x-axis, you see this beautiful correspondence to the results of our simulation. And you see how these death-birth processes mostly lead to suppressors of selection across all kinds of graph families. Now, for the birth-death process, the story is a little more complicated. So why is the story a little more complicated? Well, let's look at this plot here. So I have standard variation in degree. And let's just take the middle green line, right? Standard deviation in degree. All networks for that particular standard deviation of, 7 there for the green line, have the exact same mean degree, the exact same standard deviation in degree, and yet you see a huge range of probabilities of fixation. So this tells us that there's a little bit more that drives probabilities of fixation here. And in fact, as we compute this amplification parameter, it turns out that this amplification parameter for the birth-death process also greatly depends on the correlation in degree. So this variability of, am I connected with nodes of similar degree to me versus much higher or smaller degree, plays a large role in the probability of fixation and in the selection amplification parameter of a network for the birth-death process. And in fact, as we vary the selection amplification parameter for the birth-death process, we see that in not most graphs under the birth-death process are, in fact, not just amplifiers. You see a huge range of amplifiers, but also suppressors, even for the birth-death process, a result that wasn't really seen before. We also have a bunch of other results as they relate to how far can you push the limits of this amplification or suppression, and can you design population structures that span the two. But I'm going to skip through that because I don't want to run out of time. I want to tell you about a bunch of other things. So one application that we have found for this work is to use these microscopy images, these spatial cellular images in the bone marrow, together with our collaborator, Cesar Nobel Arrieta, and using these images that are taken from the mouse femur, they're immunostained, we can build a spatial distribution of the stem cell niches in the bone marrow because of the particular properties and the particular types of cells that create the stem cell niches. We're able, through imaging, to figure out where the stem cell niches of the bone marrow are. Moreover, while niches are stationary, stem cells can move between the niches, and we can use other studies to try to understand what migration rates might there be between these niches? In other words, how far can these stem cells move? And trying to understand how far can they move is akin to essentially building links between these stem cell niches. If the stem cell niches are nodes, the migration patterns between them are like links between these nodes. And so we can go from the spatial distribution into a spatial mathematical representation of the bone marrow stem cell niches, right? So these are two ways of viewing the same problem, right? You can build these mathematical representations through a network of movement between the niches. And then you can ask, how does the spatial structure here affect rates of mutation accumulation, for example? And using our evolutionary models, we see that regardless of the process, be it a birth-death or a death-birth process, these niches are suppressors of selection across a wide variety of parameter regimes. Now, this is, in a way, we were very relieved that we found these networks that act the same across the two different processes, because a big problem is the fact that the choice of process itself affects whether a network is an amplifier or a suppressor, right? And so it bugs you when, it acts one way for a particular process and another way for a different process. So you really need to make sure of the process. Here, these spatial architectures, regardless, and we try to, this is a result very robust to parameters, were suppressors across process, which is a very strong result. Now, what about times to fixation? I told you about probabilities of fixation. Times to fixation are yet another big part of understanding rates of evolution in a system. And in fact, previous work, again, on very small networks, I think this is a result for graphs of n equals 10, have shown that there is always a trade-off between fixation probability and fixation time. And this is indeed what we see in our result too. When we increase fixation probability, fixation time goes up with it. So the question becomes, for all kinds of different evolutionary optimization problems, I'd like to have high fixation probability, for example, but low fixation time. So is there a way to decrease time to fixation for a given probability of fixation? I want a fixation probability that I have designed that I want. How can I decrease time to fixation? And we were inspired, and I love sort of these, when you're inspired by some very old papers. So one of the papers we were inspired for this project is this paper from 1965, or more recently, a paper on the human connectome and the brain, where the authors basically make the case that networks are not just defined, or network properties that matter are not just degree distributions and mixing patterns, but also network motifs like these triangles or cliques or cavities, right? So as we move from dimension 1 node to 2 edge to higher dimensional motifs like triangles, cliques, and cavities, what role, right, do these motifs have in shaping evolutionary dynamics on this network? So for this project, the birth-death and the death-birth dynamics was exactly the same. And we were able to, again, build these edge swapping algorithms that allow us to keep the degrees distribution constant. So mean degree, variance degree, everything about the degree distribution and the mixing pattern constant, but only change, for example, number of triangles in a network. And as we do that, we have realized that triangles conveniently change times to fixation, but do not change probabilities of fixation. So as we vary the fraction of triangles on the x-axis, the probability of fixation is kept constant above the well-mixed line. We've just assigned these graphs. For example, here we're using k-regular graphs. So we've designed them to be above the well-mixed model. But the times to fixation, we can play with and change dramatically. And in fact, we find for times to fixation, we find this equivalent of the amplification factor for probabilities of fixation. And instead here, we call it the acceleration factor, this factor that tells us something about how the network changes times to fixation of a new mutant in the population. And again, our results are pretty stable to strength of selection, as well as all kinds of other parameters in the model. And I don't have a lot of time to, again, go into the mathematical intuition here, but what the crux of the proof lies in this observation that the fraction of triangles in the network essentially affects this equilibrium that we're seeing throughout most of the evolutionary trajectory of these edges that connect nodes of different types in the network. The more triangles you have, the lower this equilibrium. And we're using this equilibrium to essentially we're using this equilibrium in our analytic approach. And I'm going to skip over this because I am running out of time, but I wanted to introduce you briefly to also a slight different flavor of application that we also are working on in our group. So in addition to our image work, we are also moving a little bit into designing some of these spatial structures that help with evolutionary strategies for reinforcement learning. So you can use some of the approaches that we build here to essentially tune the rate of solution discovery across a wide variety of problems. So here in this problem, this is a well-known problem called the cart-pole problem. We use the spatial structure to evolve an agent to optimize reward. And we see that by tuning the fraction of triangles, we decrease the time until the agent reaches this reward. And this is yet another way to look at this problem. By lowering the number of triangles, we reach the reward faster. There are all kinds of other optimization problems where an increased fixation time is preferred and not necessarily a decreased fixation time is preferred. So in problems where you have very rugged landscapes, so a lot of local minima, we design population structures that start far away from the minima, and we try to understand the role of these population structures in helping the population not get stuck in a local minima and cross the fitness valley. And we see that these designed population topologies indeed do much better than well-mixed populations. And the plot now looks a little bit different, where in these types of applications, you want to increase fraction of triangles because that will help you, that will give you enough time to reach your goal. Now, this also gave us a sort of inspiration to take the evolutionary work further and instead also look at the role of spatial structures when it comes to multiple potentially interacting loci and mutations. So all previous work I've told you before is about this one new mutant appearing in a random node in a population. What's its probability and time to fixation? What if now you have, let's say, something like stochastic tunneling? You have to cross a valley. Does the population structure help you or not to cross that valley? And when does it help you versus not? So the case of, for example, a stepwise fixation would reduce to the case of one mutation model, because there you'd have, let's say, an intermediate mutant that fixes before the second mutant starts to spread, right? So you can have this stepwise fixation. However, what happens if you don't have stepwise fixation? What happens if you have an intermediate mutant starting to spread in the populations and you can get the second mutant faster than the first? And it turns out that this complex interplay between probability and time of spread for this intermediate mutant, right, for the spatial structure shapes how this intermediate mutant would spread and how long it will take it to spread. And this complex interplay leads to a lot of interesting evolutionary behaviors that do not reduce to the one mutant case. So just to give you a small little glimpse into the problem, let's say we have selection strength here on the x-axis, fixation probability on the y-axis, and this just shows what is it? A suppressor of selection, right? So we have an amplifier of selection. Sorry, we have the well-mixed pink line, and then deleterious mutants do worse. At 0, that switches. Beneficial mutants do better. Now, on the same population structure under a stochastic tunneling dynamics, we see all kinds of different crossings and different behavior that don't necessarily occur, and the switch doesn't always happen at selection strength equals 0 at the neutral case. And we set about to try to understand some of these dynamics. So you go from this sort of split in the network type between suppressor, isothermal, amplifier, towards a lot more complicated evolutionary dynamics when it comes now to understanding multiple mutants in the population. And I don't have time to go into the intuition for this, but this work should come out in a few days. We're very, very close to putting it online. So, and I'm happy to talk to you more about it. And we do this, again, systematic analysis of stochastic tunneling behaviors across a wide variety of network families. And I'm going to end there. So in this talk, I told you a little bit about some of our work using network spatial representations to understand the probabilities of spread and times of spread for one mutant appearing in this network population. And then we can take this work further to try to understand all kinds of other evolutionary behaviors when you don't just have one mutant. And in fact, there's all these further evolutionary models that this work opens up on the role of structure modifiers and spatial gradients. And we started work on eco-evolutionary interactions. What are relevant structures to consider? Can we reverse the problem and infer spatial structure from the genomic data instance? And all kinds of other problems that some of my students in the lab are working on. What I told you about here, with the exception of the last little number of slides, is all pre-printed, so you can find it online. You can email us about it. And most of this work was driven by my student YP. And hopefully next time I'll get to tell you a little bit about some of the work all my other students are doing in that photo. Thank you very much.
Okay, thank you very much, Oana, for this great talk. So I see we all have a question online by Nick.
Hi, that was really interesting. There's a lot to think about there. I have a question about the relation between the fixation probability predictions that you have and the classical sort of theory. So you find that in the birth-death models, what matters is the ratio of basically the mean square D to the square of the mean D. That was the key parameter. In the
death-birth, yes.
Sorry, death-birth, whichever way around it was, yeah. And then in the birth-death, you also have to take account of the correlation in D. And that's sort of reminiscent of the key driver of fixation probability in a population where individuals just have varying fitness is the same ratio. It's the variance of fitness relative to the mean. And you also then have an effect of the correlation in fitness across generations. So I'm making an analogy between number of offspring and degree, but I have no idea why there'd be a difference between the birth-death and the death-birth in this case.
I think that's a great point. I think we're also actively trying to sort of put these results together, kind of like you were saying. Let's try to wrap our heads around how these results fit when it comes to, let's say, more traditional results, and also results that seemingly may seem different, where you have kind of like exactly like you were saying, you seemingly may have differences in selection, but really maybe it's about the number of nodes you're connected to, because that's where you can leave offspring, right? So we are actively working on that, and we're trying to understand why exactly you see these differences, what the results are. But we're still sort of these are very new results for all of us, and we're in the process. I mean, we're trying to build, in a way, simpler models to really try to create these very explicit parallels. But I will have an answer for you, just not right now.
Okay, thank you.
Mm-hmm.
Okay, some other questions? Vincent?
Hi, Oana. Thank you very much for the nice talk. It's Vincent Calvez speaking. I have maybe one very short clarification and then one question. The first point is about the application you mentioned. Did you say that your result does not depend on the stochastic process beyond, whether you do first birth or death? Is it what you got?
Yes, okay. Because as we saw, for example, in this plot, let me just quickly go back. Oops. Sorry. I don't know why it's not reacting to me anymore. But essentially, in the birth-death plot, you saw that we found this result where you now can have networks that are also suppressors of selection. And it turns out that these are particularly spatial, like one category of networks are these spatial, either normal geometric or uniform geometric. And in these spatial, as we build the networks of the of the stem cell niches in the bone marrow, we use some of these spatial networks because either way, either be it deterministic or probabilistic, we connect nodes proportional to distance. And specifically for these types of spatial networks, the two processes end up being very similar. Now, for let's say networks that are used in different types of processes, like a cultural evolution process, let's say a PA graph, right? Preferential attachment graphs. Those have very different expectations across parameters for birth-death versus death-birth.
Thank you. And my second question, maybe a curiosity question. I noticed in one of your last slides that there was some maybe non-monotonicity behavior with respect to the population size, so the parameter NS. That was in one of the last slides that you.
For the one mutation, or
I think it was one or two slides before this one, if it's possible to. Yeah, in this one you have some strange behavior on the right top right corner with respect to the population size, which seems to be specific to this which is not to be the case with only one mutation dynamics. So which seems to be that when you increase population size, you have a non-monotonic effect. I was curious to see if it's something expected or something surprising or something that can be a match to some standard.
Right. So here we're plotting NS, and the black line is at NS equals 0, right? Because that's when the switch happens in the case of one mutation. And indeed, as we vary the strength of selection, we do see these different behaviors. And here we've almost colored them red or blue based on, does it almost act like an amplifier or a suppressor? So you get this change between, let's say it's a suppressor because it amplifies deleterious mutations, then it switches to an equivalent of an amplifier because it amplifies beneficial mutations, but then it also suppresses beneficial mutations, and so it switches to a suppressor again. So we're trying to understand some of these different weird-looking behaviors that happen in the case of stochastic tunneling. And we do see all of these different classes of behaviors that we're trying to wrap our heads around.
Thank you very much. That was very clear. Thank you.